Hello guys, welcome back to my channel. The topic of my today's presentation is staining technique in microbiology. What is a staining technique? First of all, staining technique refers to a method used in microscopy to enhance the contrast of microscopic structures or cells by applying colored chemicals. By saying colored chemicals, we are referring towards dyes or stains. So these process helps to highlight specific components of specimens such as the cell wall, nucleus or organelles, making them more visible under a microscope. So in simple words, a staining technique is a method which is used in microscopy to enhance the contrast of microscopic structure in order to view a microorganism in a proper manner, in order to view its structure, size or shape or its morphological details. We use the staining technique. So staining technique also have different types. There's simple staining, there's differential and special methods of staining. Moving on to the next slide. Why we stain bacteria? First of all, before studying about staining technique, or before doing a brainstorm regarding staining technique, we need to understand that what is the need of doing staining. Why do we even need to stain bacteria? There must be various reasons behind doing it, because nothing in this world exists without a reason. So first of all, why we stain bacteria? The very first reason of staining a bacteria or any microorganism to view it under a microscope, whether it's a bacteria, whether it's a virus or any other microorganism, the very first reason is bacteria are naturally transparent and colorless. Visible enhancement, to enhance its visibility, we stain certain microorganism. Moreover, we really need to keep this thing in mind that bacteria are naturally transparent and colorless, making them nearly invisible under standard light microscopy without staining. Imagine any researcher, any scientist studying about a particular microorganism and he wishes to view it under a microscope. Now, if this particular person do not stain the particular microorganism, he won't be able to view it under a microscope. No matter how expensive that particular microscope is, no matter how old or no matter how ancient that microscope is, or no matter how advanced that microscope is, the bacteria needs to get stained in order to get a view of it, because bacteria are naturally transparent and colorless. We can't view any bacteria under a microscope without staining it. So staining is an essential part in order to visualize microorganism. The second reason of staining any bacteria is morphological details. As I said, staining also reveals cellular structure, shapes, and arrangement that are crucial for proper bacterial identification and classification. Without staining, we won't be able to identify the shape, structure, or the arrangement of any bacteria, because nothing will be possible under a microscope. If you just grab a certain bacteria and put it under a microscope, you won't be even able to identify a single thing, because bacteria is transparent. Everything will just look clear and transparent. You won't be able to view it unless you stain it properly. The third reason is diagnostic precision. Different staining properties help distinguish between bacterial species, guiding appropriate treatment decisions in clinical settings. As I said in my previous video also, there's various microorganisms present on earth, and in order to differentiate each microorganism, we need to stain these bacteria to differentiate which microorganism is responsible for causing which particular disease. Otherwise, we won't be able to even recognize any bacteria, recognize any disease or bacteria, or even find any treatment or prevention of any disease, because we won't be able to link things between them, between a bacteria and disease, and we won't be able to identify which bacteria is causing this certain type of disease. This is also one of the most important reason why staining is important, in order to differentiate the properties between bacterial species. There are various kind of bacterial species, and we really need to differentiate between them. So the differentiation can only be done by reading their morphological details. Otherwise we won't be able to understand which bacteria is which, or what bacteria is doing what, or how can we differentiate, or how can we name certain type of bacteria. So these are the three main reasons why staining is important in microbiology. Moving on to the next slide. Simple stain. Now, what is a simple stain? Do anybody of you know what is simple staining? So simple staining is a staining technique that uses a single dye to color all cell or structure in a specimen. So simple staining is one of those techniques where only a single dye is used to color all cell structure in specimen, primarily to observe shape, size, and arrangement. Its purpose is to visualize basics of cells. Like the dye which is used in simple staining is the common dye which is usually used in simple staining is methylene blue, saffron, and crystal violet. These three dyes are usually used in usually are usually used while performing simple staining method. So simple stain, single dye application that color all bacterial cells uniformly, providing basic morphological information. If you are interested in getting any in grabbing any particular or basic knowledge about a certain microorganism, you only need to do a simple staining method just in order to visualize or get the morphological information of any cells. So you can use these dyes: methylene blue, saffron, crystal violet. Methylene blue will help you. It's a basic dye with positive charge. It is excellent for cell shape visualization. If you are very much interested or enthusiastic about identifying a certain shape of a certain bacteria or certain virus, you just take methylene blue, and in order to visualize certain shape of bacteria, you can use this dye on a glass slide and then view it under a microscope. But you need to do it in a very proper manner, not just you can't just simply go in the lab and start doing random things, but there is a proper process of doing even a simple staining method. But this is a dye which is used in simple staining method. So this is methylene blue, which is safe and easy to use. Then comes saffron. It is a red basic dye commonly used counter stain, provides accent contrast. Then comes crystal violet, which is primary stain in gram method, deep purple coloration, highly affinity for cell walls. Also, crystal violet is used in gram staining to differentiate between gram positive bacteria and gram negative bacteria. Moving on to the next slide. Negative staining. What is negative staining? Negative staining is a microscopy staining technique in which the background is stained, but the cell or the structure remains unstained and appear clear or bright against a dark background. So it is a unique approach where the background is stained dark while bacteria remain unstained and appear bright against the colored background. So in simple words, if I say, what happens, negative staining is mainly used for microorganism those which is delicate, and also for those organism which is hard to stain. So it is negative staining is usually used for two types of microorganism. Number one, which is delicate while staining, and which is hard in the sense when it's difficult to stain it. So it is negative staining is used for these kind of microorganism. The first one is the for those microorganisms which is delicate in nature, and second which is hard in nature. So yeah, India ink method creates dramatic contrast by staining the background black. Bacteria appear as clear, bright cells surrounded by dark medium. Ideal for capsule demonstration. No heat fixation is required and preserve natural cell morphology. Then comes necrosin application, which is alternative negative stain producing gray black background, particularly effective for visualization, bacterial capsule, and cell boundaries. Less intense than India ink. Oh, it's a India ink, sorry. Better for photomicroscopy and excellent capsule visualization. Moving on to the next slide. Then comes differential stains overview. What is differential staining? Till now we studied about simple staining, then we studied about negative staining. Now what is differential staining? Differential staining is a technique that uses two or more dyes to distinguish between different types of cell or structure based on their staining properties. Let me repeat it for you once again. Differential staining is a technique that uses two or more dyes to distinguish between different types of cells. Now, differential staining, as I said, there are many staining techniques. Not only simple staining techniques is available in the labs, but there is differential staining technique where two types of dyes are used to distinguish between different types of cells or structure based on their staining properties. So, distinguished bacteria groups is primary stain. Initial dye colors all bacteria cells uniformly. Then there is decolorization, selective removal of primary stains from certain cell types. Then there is counter stain, secondary dye provides contrasting color to decolorize cells. Then there is classification, final color distinguish between bacterial groups. Okay. Now, what is the purpose of differential staining? Differential staining is to differentiate between groups or components. Like, for example, gram staining is a differential staining method. If you would like to differentiate between certain type of microorganism, for example, if you want to differentiate between a gram positive and a gram negative bacteria, over here we will use differential staining method. Differential staining method, the purpose of differential staining method is to differentiate between groups or components, and the best example of differential staining method is gram staining, where you distinguish between gram positive and gram negative bacteria. The first example of differential staining is gram staining and acid fast staining. Let's move on to gram staining. Now, what is gram staining? Guys, please don't get confused while studying about different staining techniques. It's a very simple thing. When you just read it 2-3 times, then you will understand it everything. It's not rocket science, so it will be very easy for you to understand everything about it. Just you need to revise it properly and keep the main things in your mind. So what is gram staining? Gram staining is a differential staining technique used to classify bacteria into two major groups, gram positive and gram negative, based on structural difference in cell walls. So, the gram staining, the gold standard for bacterial classification. Number of primary strain, as I said, crystal violet stains crystal violet stains all cell purple. Then there is mordant application, which iodine fixes the dye to cell walls. Then there is decolorization. Well, alcohol removes stains from thin wall cell. Then there is counter stain, saffron colors decolorize cells pink red. So then, okay, now the purpose of gram staining is to distinguish bacterial species by the composition of the cell wall, primarily the peptidoglycan layer. Also, the principle of gram, just a second guys, I think there's some sort of glitch. Just a second. Yeah. Also, the gram positive bacteria have a thick peptidoglycan layer that retains the crystal violet iodine complex appearing purple, and gram negative bacteria have a thin peptidoglycan layer, an outer membrane that do not retain the violet dye, instead take up the counter stain safranine appearing net. So guys, you need to keep in mind the purple The purple is the gram positive bacteria, which are the bacteria which is appearing purple in color, is it retains the crystal violet iodine complex, and it starts appearing purple. So that one is the gram positive bacteria, and the one which appears to be pink or red is a gram negative bacteria. And how we differentiate or how we identify, there is one more way of identifying it, by its peptidoglycan layer. The gram positive bacteria have a thick peptidoglycan layer, while a gram negative bacteria has a thin peptidoglycan layer. And we need to keep these two things in mind. Also, the reagents which is used in gram staining method, crystal violet is the primary stain. It's already mentioned over here. The primary stain is the crystal violet stain. Then there is iodine, which is the mordant, form complex with the crystal violet. Then there is alcohol, which uses for decolorization. Then there is counter stain, which is where safranin is used. Moving on to the next slide, acid fast staining. What is it? Is acid fast staining? Specialized technique for identifying bacteria with waxy lipid-rich cell wall that resists decolorization by acid alcohol solution. Now, what is acid fast staining? Acid fast staining, in simpler word, is a differential staining technique used to identify bacteria that have waxy lipid-rich cell wall. So over here, acid fast staining is basically used to acid fast staining is a differential staining technique used to identify bacteria that have waxy lipid-rich cell wall, which make them resistant to decolorization by acid alcohol. So, target organism: Mycobacterium species including M. tuberculosis, leprosy, and atypical mycobacteria. Nocardia species, partially acid fast, important in immunocompromised patients. So, what is the purpose of, in simpler words, the purpose of acid fast staining is to detect acid fast organism, especially Mycobacterium species. If you want to identify Mycobacterium tuberculosis, which is responsible for causing tuberculosis in human beings, which type of staining methods can we use, or which type of staining method is used till now in labs? It is known as acid fast staining. Acid fast staining is responsible for detecting acid fast organism, which also includes Mycobacterium species. Mycobacterium tuberculosis is an acid fast organism, and this can only be detected by acid fast staining. This is the reason why we use this. Then there is clinical application, TB diagnosis, essential for rapid tuberculosis screening in sputum samples, leprosy detection, critical for identifying M. leprosy. Also, acid fast staining is also used for detecting leprosy. So till now, the two essentiality of using acid fast staining is to detect tuberculosis in sputum, and number two, in the detection of leprosy. Then comes staining's result. Acid fast bacteria retain red carbol fusion stain after acid alcohol treatment. Non-acid fast appears blue from methylene blue counter stain. Moving on to the next slide. Special stains for bacteria structure. Now there is a special stain. There is also special type of staining technique which is used for bacterial structure. Till now we studied simple staining, we studied negative staining, then we studied differential staining, then we studied gram staining, then we studied acid fast staining, and each staining has its own importance in the field of microbiology. Without them, I think things will be completely impossible. But there's also special technique there's also special staining, and special staining refers to a group of staining technique used to highlight specific structure or components of cell tissue microorganism that are not easily visualized with routine stains like gram or simple stain. You see, the more you try studying about microorganism, the more it gets complex. Right now we study different kind of staining techniques, but in the last we have special staining technique. And special staining technique refers to the group of staining technique used to highlight specific structure. Now, for highlighting in order to identify specific structure of the component of cell tissue microorganism that are not easily visualized with routine stains like gram like gram or simple stain, then over here a special staining technique is used. Now what is the purpose? To detect visualize structures such as capsules, endospores, flagella, cell walls, fungal elements. For to detect these specific structures or visualize the specific structures of capsule, endospores, flagella, a special staining technique is used. Now let's read it. Spore stains, Scaffer-Fluton method. Malachite green penetrates spores with heats. Safranine counterstain where vegetative cells spores appear green, cells appear red. Then there is flagellar stains, which is Lefson's method builds a flagellar thickness with moderns, making normal invisible flagella visible under light microscope for motility studies. And there is capsule stain, as I mentioned before. The purpose of special staining is to detect capsules, endospores, flagella, cell walls, fungal elements. So Anthony's method combines positive negative staining. Capsules appears as clear halos around dark stained bacterial cell. Then there is advanced staining techniques. Also there's advanced staining techniques which includes fluorescent and immunofluorescent method. Fluorescent staining, and if we do side by side comparison between fluorescent and immunofluorescent application, fluorescent technique use DAPI staining, which is blue fluorescence, for DNA visualization. Then there is calcofluor, wide chitin and cellulose detection. Then there is live dead assay, viability determination. Then if we come into immunofluorescent application, there is direct IF antibody conjugated with fluorophores. Then there is indirect IF secondary antibody amplification. Then there is species identification, highly specific detection. Then there's antigen localization, which where precise cellular mapping. So these advanced methods offer super sensitivity and specificity compared to traditional staining, enabling rapid pathogen identification and antibacteria susceptibility testing. So guys, in this video, we came to know about different staining technique. We began from a simple staining to negative staining. Then we came to differential staining, which included gram staining to acid fast staining. Then we also studied about special staining technique, and right in the end also we had a glimpse of advanced staining technique. So choosing the right stain. Decision making framework for optimal staining technique selection based on clinical needs and suspected organism. Confirm required info, morphology, classification, structure. Select stain, glass, simple, differential or special. Suspect organism type, bacteria, mycobacteria, or others. Access clinical need, morphology was a classification. Okay, so as I said, we studied about different staining techniques today, and all of these staining techniques are very essential in microbiology or in any lab where you are interested in visualizing different kind of microorganism or different kind of cell. You need to know everything about these staining techniques. Also, every staining techniques is important, and if you want to be an expert in this techniques, you need to know each of these techniques very well, and you need to at least remember the basics of this staining techniques. And with good practice and observation, you will be an expert. Also, your theoretical knowledge should be very nice. With practical knowledge, you need to enhance your theoretical knowledge as well. So that's all for today. Stay tuned for the next video. If you like, please like and subscribe my channel. Thank you very much, guys. Have a good day.